Hello, AP Biology students. We're going to go over the 11th section in Unit 3. And in this section, we're going to talk about something called the light-independent reactions. Remember that we're still talking about the process of photosynthesis that takes place in chloroplasts. We already reviewed what the light-dependent reactions are. They take in light, water, and produce oxygen, but they also produce these products of ATP and NADPH. These two products are going to fuel and energize the Calvin cycle, otherwise known as the light-independent reactions. This is again where we're going to take in CO2 and produce sugar molecules. And again, this occurs in the stroma of the chloroplast. Basically, we're going to be looking at a series of linked enzymatic reactions and how they incorporate CO2 to make sugar. I know it seems like a lot, and it is kind of a lot. These are the three main parts of the light-independent reactions. They're carbon fixation, carbon reduction, and the regeneration of RUBP. Now, I'm going to be looking at each individual part a little bit more specifically, but remember that these cycles don't actually turn like a cycle in the chloroplast. We just represent them as a cycle to help us understand the process. All right, the first part, carbon fixation, and like the name implies, we're going to be fixing carbon into other molecules. A single CO2 molecule, as you can see here, is going to be attached to a 5-carbon molecule called RUBP. Rubisco is the enzyme that's actually going to fix this carbon into this molecule. Now, you might remember, hey, most enzymes end in ACE. Why doesn't Rubisco end in ACE? Well, it actually does. Rubisco is just the nickname. Rubisco stands for ribulose 1,5-biphosphate decarboxylase. But again, this enzyme just fixes that carbon molecule onto that 5-carbon molecule here, and it's going to make a 6-carbon molecule. Now, that 6-carbon molecule is not stable, so it's going to break down into 2 3-carbon molecules. I forgot to remind you that in these processes, we really only care about carbon. Yeah, there are other hydrogens and oxygens here, but we want to focus just on the amount of carbon that's being fixed into these molecules. One interesting part about this reaction is Rubisco actually fixes this carbon atom that it's getting from the CO2 in the middle here. You can see it's the 3rd carbon in. And whenever this 6-carbon molecule breaks down to 2 3-carbon molecules, you can see that position of the carbon. Now, it looks like it's different here, but it's just signifying where that carbon is attaching itself during this process. An interesting fact about Rubisco is it's the most abundant enzyme on Earth, partly because it's found in all plant life, and it just takes a long time for this reaction to occur. So plants have a lot of this enzyme, and there are a lot of plants, most abundant enzyme. Now our next step is carbon reduction. We're going to reduce the molecules here that we were just talking about. 2 reactions are going to take place. ATP and NADPH are going to fuel these processes, and remember these come from the light reactions. There are other processes and enzymatic reactions that go along in this step, but we just want to focus on these molecules that are known as 3-PGA being turned into something called G3P. Just a side note, if you ever see G3P or PGAL, they are the same molecule, we just have 2 different names for them. So again, 2 of these 3-PGAs are changed into 2 G3P molecules. Our next step is the regeneration of RUBP. Now, before I go anywhere else, one of these G3P molecules is going to go on to make glucose. We're going to talk about that G3P later. The other G3Ps are going to be remade into RUBP using ATP. Remember, there are just enzymes working within this whole step. We don't need to focus on those. We just want to understand that these G3P molecules, which are very versatile, they make a lot of other different molecules, they are going to go and be recycled into the RUBP. This RUBP, if you remember, is going to start off this whole process. This is why this whole thing is a cycle. If we look back here at the 3 different parts, you can see the RUBPs bonding with the carbon atom to form this 6-carbon molecule that breaks down to 2 3-PGAs. These get changed into 2 G3Ps. One goes to make a glucose, the other gets regenerated into the RUBP. Now, remember how I said we're going to talk about this molecule G3P and how it goes to make glucose? Well, remember, it only has 3 carbons and glucose has 6 carbons. So we're going to need 2 of these G3P molecules to actually go and make 1 glucose. That means this cycle has to occur twice since we need 2 G3Ps. Now, I kind of show my students this version of the cycle and say, hey, look at the carbons. Where is there a problem? And a lot of my students say, hey, there's only 3 carbons here, but there's 5 carbons here. Where do we get those extra carbons from? Well, we got to do a little math. There are actually 3 RUBPs to begin with, 3 CO2s, and then 3 of these molecules. So basically what we have to do is we have to triple everything here. And this should make sense. We're only taking 1 carbon in per carbon dioxide. So we need 3 of them to make 1 G3P. So it kind of looks like this. 3 carbons of carbon dioxide are going to be attached to 3 RUBPs. They're going to make 3 of those 6 carbon molecules that make 6 of these 3 carbon molecules. They are going to be changed into G3Ps, one of which goes to make glucose, the other 5 go to make RUBP. There are 5 3 carbon molecules here that are going to be recycled into 3 5 carbon molecules here. And then this process starts over again. So this is actually what it looks like further when you have this cycle occur twice. Now, I'm not going to go through every part here, but you can see we're intaking 6 carbon atoms through 6 CO2s just to make 1 glucose molecule that has 6 carbons. And that G3P molecule that we were just talking about is a very versatile molecule. It's a starting molecule for a lot of other organic molecules, such as glucose and other molecules just in general. You can see here it makes cellulose, starch, plants oils, and just a ton of other organic molecules for plants.